So what we're going to do today is cover a few aspects of the EuPathDB resources, and we will focus on FungiDB, and also we'll use PlasmaDB. Although, as you know, EuPathDB, I'm showing the front page of the EuPathDB portal site, and you'll see all these little icons everywhere, each representing one of our different databases that we support. So as you can see, there are many different organisms that fall under the EuPathDB umbrella. And in fact, this list is incomplete. There are a couple of additional databases that are not listed here, and these include, and I will just open them here as we speak. These include HostDB, which is a database that integrates host response to pathogen infection, and so that's an expanding database and a new resource that we've been developing over the years. Another resource is clinepidb.org. This is a database that provides access to clinical study information. And so there are a number of studies. There's one that is available now publicly, and a number that are coming up in the near future that will become available. And these are very different from, if you're a molecular biologist, somebody who's used to working on genomes and genes. This resource is very different because it allows you to look for participants in a study based on various criteria that were collected over, let's say, a 10-year period and so forth. And this is a new resource that is, I think, a very exciting portion of the databases. An additional resource is microbiomeDB.org, and this is a resource that includes microbiota information. And so there's a number of studies. There's a new release coming out very soon that'll include even additional studies where you can start asking questions about the microbiome or microbiome studies. And so, what is the bacterial population in the gut of patients that are, treated with antibiotics or so forth. And so various different types of questions that you can ask here. As the years progress, these resources hopefully will become more and more connected. You can imagine how a host resource can be, more integrated with a parasite resource or a pathogen resource, where you can start asking integrated questions between the host and the pathogen. But also you can imagine now expanding this to other aspects, whether it is identifying pathogens in a particular clinical cohort, and then drilling down into the pathogens a bit more if you have, let's say, whole genome sequencing of all the pathogens, and you want to look at the types of polymorphisms or variants that are available in these pathogens that came from certain participants. And the same thing would apply to microbiome information. So regardless of whatever resource you're using, they're all pretty much built on the same infrastructure, and the tools that are available to you, obviously depending on the type of data, are the same or very similar, sort of following the same philosophy, and that is to allow you, the user, to ask your own questions of the underlying data. So the goal is not to just recreate the data or the figures that are in a paper, because that's why the papers are there. The goal is really to give you access to the underlying analyzed data most of the time, where you can then ask your own questions by modifying the different parameters in the searches. And as we show you some examples of searches today, you will hopefully appreciate the power of being able to do that. In terms of the layout of our sites, so it's very basic. If you've used these sites, I think all of this will be redundant to you, but if you haven't, hopefully you'll learn something new here. But all of our sites contain a banner section, so it doesn't matter if you go, to GRDB, let's say, it looks essentially the same. The colors are different. We have this banner section on the top, which includes the logo of the resource, an ability to search for gene IDs or by text. And so, but these are searches that return genes. There's a number of helpful information here about the database. Various access to various help information, ability to log in. I'll tell you why it's important or useful to log in. And to log in, you have to register. And so there's a quick link here to register. The contact us link, we already told you about this. So this is a very useful way for you to be in touch with us and let us know what you need. and then access to various social media, including Twitter, Facebook, and YouTube. We have a number of YouTube tutorials available to you. In fact, this webinar today is being recorded, and we will make it available. You will get a link for this webinar afterwards, but also we will make it available as a YouTube tutorial for others to be able to access, and for you as well to access later on.
Amar, and just a quick thing to add, we did post a handout which is available for you to download. The handout contains link outs to various tools, including our YouTube channel, where you can watch the videos.
Great, thanks, Eve. This gray bar right here includes basically access to all of our searches. So you'll notice here that there are drop-down menus, and you can access all the searches. One thing I think you will notice in our sites, there's lots of redundancy, and that's because people are used to accessing things from different places. So for example, all the searches that are available here under New Search with all of these tiered menus are also all available right here in the central portion of the site, and that applies to all of our resources. There are some aspects available, tools available to you from the gray menu bar that are not necessarily available elsewhere. For example, the ability to analyze your own experiments, and this is using the Galaxy platform. I know Marcus is very familiar with that, and we will. We're not going to go through details of Galaxy today, just because of the time. We can spend an entire day just working on Galaxy, but what we will do is show you a demo of a new feature that has been made available to be able to integrate data that you've analyzed in Galaxy and take it into another section of our resource called My Datasets, which is a page that allows you to integrate analyzed data into the resource, so you can privately analyze your own primary data. We have this banner here. As you'll notice here, there's an announcement for a fungal genomics workshop. So if you're a fungal biologist and you're interested potentially in attending a workshop, this one is held in the UK. We would encourage you to apply. There's information available by following the link up here. It's a pretty nice workshop, actually, covering multiple resources, and they do offer various types of bursaries. So I would apply and not worry about the money for now, and then think about the money afterwards. But if you don't apply, obviously there's no chance of going, regardless of whether the money is available or not. Scrolling down, this left menu section again offers you many different aspects, or access to many different aspects of the site. For example, a data summary page, which shows you all the genomes that are available in the database. So if I click on this, this will run a search, essentially, that returns all the organisms in the database. And you see here that there are 330 organisms in EuPathDB. And those are both annotated and unannotated genomes. And they're listed below here with a lot of information about what is available for each of the genomes. And so again, this is a nice way to quickly go back and see what is available in the resource. Our Twitter feed is listed here. If you go to FungiDB, you'll notice that we have not only the EuPathDB Twitter feed, but also the FungiDB Twitter feed. And so this is a quick way to provide you with information. There are additional community resources down here. What I would like to point you to is the education and tutorial section right here. And this section has access to the YouTube channel, but also our workshops or exercises from our previous workshops. And so, for example, you can go and see the exercises from our most recent workshop at the University of Georgia. So this is another workshop we annually run in Athens, Georgia, and the applications actually will come up soon, and so something to look out for. This is a general EuPathDB workshop, and again, we encourage people to apply to this one, and this takes place usually in June in Georgia. But if you click on this, it'll take you to the workshop page, and this is the schedule, and you can see here, these are all the different exercises that were run during this workshop, and each one of these will link you to a PDF, which includes the exercise that was used during the workshop. So this is another useful resource for you to look at. You could go to our general workshop page, and so if you just go and visit all of our EuPathDB workshops, this lists all of our workshops. And you'll notice here that there are a number of them that were run in a couple in 2018. There's actually one more that needs to be added here. but this is an expanding list of exercises. Obviously, the old ones will become slightly obsolete because the resources and the type of data that's in them is updated over the years. Okay, as you noticed, I'm now in FungiDB, and so I'm going to just demonstrate one quick search and show you how these searches work, and then I think we will slowly then turn it over to Eve, and she's going to cover other aspects of the site. But what I will start with, I'm just going to start with a quick text search right at the top here. So I'm looking, let's say, for genes that contain the word kinase in them, because I'm interested in kinases. And so I'll just type the word kinase up here at the top right-hand side and click on the search icon. And so this will return any gene in the database in all of FungiDB where the gene contains the word kinase somewhere in its record. So the gene record is very rich with a lot of information, including the name of the gene, but also information from the various types of curation, for example, Go terms or interpro domains, but also information that's coming from our users, so user comments. We'll touch on user comments at some point, hopefully today. It includes information about where the genes are expressed, if there's expression information, there's structural information, similarity to the protein data bank. It also includes a lot of other useful information about the gene that's not necessarily in the name of the gene. So a gene could be hypothetical, and that hypothetical gene may actually have an interpro domain that has a kinase domain in it, and that gene would be returned by simply running this search. And so, as you can see, this search took a little bit longer. It searched, hundreds of thousands of records to find this, but it found 48,461 genes across all of the fungal organisms in FungiDB that contain the word kinase somewhere in their record. And as you can see here, there's a table. We call this the organism filter table. It's currently just alphabetically listed, not the best way to do this maybe, but we're working on a better filter table that's more phylogenetically relevant. But as you scroll down, you'll see here that these 48,000 genes fell into 5,754 orthology groups. Okay, so many of them are orthologs of each other, so that's not surprising. And as you go down, you can see the various fungal groups and phyla and then species, and you can scroll to the right, and you could find your specific species of interest. So if you were, for example, interested in Aspergillus nigilans only, and you don't care about the rest of them, you can just click on this little number here, 432, and this will filter your results. It'll show you a little filter icon right here, and it'll reload your results. These are now your 432 genes that are annotated with the word kinase or have the word kinase somewhere in their record from Aspergillus nigilans only. The results, so these 432 genes, which are shown here as a step in what we call a strategy. So this panel here is your search strategy panel. And it's really like a online or in silico experiment, essentially. So imagine this is your test tube, and you can start doing different experiments in this test tube, adding different things, asking different questions about your results. And I'll demonstrate this in a second. As you scroll down, you will notice I showed you the filter table already, and you'll notice now that these kinases are all listed here. Each row is one of the genes. And it tells you where the word kinase was found. So for example, for these genes, this gene, for example, the first one we're looking at, the word kinase was found in the PubMed ID, in the record, Go terms, phenotype, interproscan, in the product description, which we can see it right here. It says kinase right here. And also there's a field called notes in there. So there are multiple sources where the word kinase was identified. As you scroll down, you'll undoubtedly find some that do not have the word kinase in the product, and those are quite exciting because those are potentially, things that are not entered as kinases, but still potentially function as kinases. As you'll notice here, there are additional tabs. So, for example, we can look at the distribution of these results on the genome and So depending on a number of results, this may take, oh, it wasn't too bad actually. So you'll see here, this is all the chromosomes from Aspergillus nigulans, and the genes are marked here in blue or red for forward or reverse strand. And you can see basically a distribution of kinases amongst all the chromosomes. You know, with a quick, without obviously doing any detailed analysis, you could argue, oh wow, look here at for some reason the left arm of chromosome 6 does not have any kinases on the reverse strand. You know, whether this is biologically meaningful or not is obviously, I don't know that, but that's just a quick and dirty way for you to see if there are any general distribution of the genes. Depending on the organism you're looking at, there may be some clear distribution of genes towards the subtelomeres, for example, or clusters. You could see some of the secondary metabolite clusters sometimes in here. They're, you know, and they will show up as a thicker line here. So, for example, this one represents multiple genes in this area. And if you click on this, this will show you the number of genes. So there's all of these 1, 2, 3, 4, 6 genes that are found here in the same particular area. These are all potentially kinases, and so that's kind of potentially of interest and something that somebody would want to follow up on if they work on kinases in Aspergillus nigulans. They may in fact be part of a secondary metabolite cluster. Another tool that we will discuss is analyze results. I think Eve will cover this at some point, but you can take results that you have and run various enrichment analyses on them, and we will discuss them in a few minutes. So back to the gene results. As you can see here, there's this table is essentially, imagine this like an Excel table. So you can close columns. So the score, for example, this is used really internally. You don't really have to worry about it. So I'll just go ahead and close the score, for example. And what will happen, it'll just remove this column from my results. But you can add additional columns. So I can click on Add Column, and I could say, well, I really also want to add the gene name, for example. And so I'll click on Gene Name, and then I will click on Update Columns. And then this will add an additional column here that includes the gene name as well. And depending on the organism, the gene name may be available or it may not be available. You will notice for some columns, there will be a little graphic. This graphic represents also a way for you to quickly analyze the results in this column. And so if I click on this, this generates a word cloud that shows the words in the product descriptions. And so you'll notice that protein is the most common word, so not terribly useful, but you can filter those out. So I can take, for example, I want to start with number 2, and so that will should filter out so took out kinase, and you can distribute these also by alphabetical or just by rank order, depending on how you want to look at this. But clearly the word kinase is the most common word in the product descriptions, which is not surprising because that's exactly what we searched for. And you can mouse over this and it tells you there are 226 occurrences of kinase in the product description out of these 400 or so results. Okay, so another, as I was mentioning, there's this strategy panel here is essentially an experiment. We're starting an experiment, right? So we found a bunch of kinases that are or potential kinases. In our case, we filtered on Aspergillus nigulans. You may be interested in asking, well, how many of these kinases are potentially secreted? So let's assume there are some important kinases that are secreted, and so, or you're looking for those. We don't know the answer yet, but we can add a step. So you'll notice here this red button right here. So this button is called an Add Step button, and this allows you to add additional searches and combine the results of those additional searches with our first result here, which was the kinase search. So I'm going to go ahead and click on Add Step. And once you click on Add Step, this generates a pop-up which has access to all of the searches within the database. In our case, I'm just going to ask for another gene search. And this reminds me to highlight something that I forgot to mention on the homepage, and I'll get back to that in a second. But I will run a search for genes, and in my case, I'm interested in genes that are secreted, and these are different categories, right? All the searches are categorized, and so obviously, depending on how you view secretion, you may assume it'll be present in different categories. We chose to put it under protein targeting and localization, And then as you click on that, you'll notice there are two options here. One is predicted signal peptides. So this just uses the signal P algorithm to look for potential signal peptides. There's also a transmembrane domain search, which we can add to our search if we wanted to. So as I click on predicted signal peptide, now you get an option here. The first option is to choose which organism you want to search for this. I could select all, right? And then this operation, when I do my intersect, it'll take care of making sure you get the right results. Or I can ask for specifically the genes from Aspergillus nidulans, in which case I can clear all and quickly here. If I expand everything, you're going to have to try and sort through and find your organism of interest, or I can just quickly start typing, nidulans, and you'll see here that it shows up right here. So autocorrect, there we go. And so now I can select it, and so now I've selected Aspergillus nidulans, and I can ask. So the question now, this search is going to find all genes that have a predicted secretory signal peptide in Aspergillus nidulans, and once it finds those results, I have to combine the results, the secreted protein result, with my kinase results, which is right here in the background. And so you have different options to choose. Intersect, which will give you the IDs, the gene IDs that are in common between two lists. But then you have other options here, like a union and minus operations. And there's also a co-location operation, which I don't think we're going to discuss today. But if people are interested, this is a way for you to co-locate entities on the genome. So let's say you have single nucleotide polymorphisms, and you want to co-locate them with genes. So let's say you want to find, let's say you identify 1,000 polymorphisms, and now you want to ask, give me the genes that are located within 100 nucleotides of those polymorphisms, for example. And so this co-location, it's almost like using the GPS coordinates of your genes and your SNPs to co-locate them. So it's actually a nice way to find things on the genome. In any case, so this search will identify signal peptide containing genes. There is a section here for advanced parameters. So if you're ever interested, you can go in and actually really tweak the knobs a bit more. And you can, we use two different algorithms for predicting signal peptides. One is a signal peptide neural network, and the other one is a hidden Markov model. And so you can, and we're combining, we're bringing in the results from both of these algorithms. You can make this more stringent and say, well, it needs to be present in both algorithms or any. You could also increase the values here to make it more stringent. I typically leave these at the defaults because they work well, but of course, depending on what you're doing, you may choose to change these. Okay, so in the end, once you're satisfied with how you set up the search, you click on Run step. And so now it's searching for all genes in Aspergillus nidulans that have a predicted secretory signal peptide. So now you'll have a two-step strategy, not a single-step strategy. And you'll see here that it found 1,846 genes in Aspergillus nidulans that have a potential secretory signal peptide. 51 of them are also annotated or were in the same in the list of genes that had the word kinase associated with them, right? So presumably right now we've identified 51 secreted kinases in Aspergillus nidulans. So an additional powerful tool in the databases is a tool that allows you to leverage orthology, so to go across different species. So let's say I'm interested in these kinases, but also I'm interested if there are orthologs of these kinases, these secreted kinases, in other organisms in FungiDB. So I can add a step, and we have an option here to transform by orthology. And so I can click on transform by orthology, and now I can select, well, which organisms I want to find orthologs in, and I could actually just select everybody, right? I could say, well, I want to look for orthologs across all organisms in FungiDB. And so we can run the step. And so now this is going to look for, it identifies any orthology group that contains one of the genes in our 51, and it returns all the other fungal genes that are in that orthology group that are in FungiDB. Okay, so this may take a couple of seconds, but ultimately it should run. So while this is running in the background, I am going to open up a new page here. And let's go back to FungiDB. I just wanted to show you the home page. Something that I forgot to mention is that the types of searches that we have are, let's see if this one ran here. It's still running. Okay. All right, we're experiencing a little bit of slow connection here. Let me see if I still have this open. So I have another page here, PlasmaDB. So again, as I mentioned, it doesn't really matter which website we're looking at. I'll wait for the other things to load. But one thing I wanted to mention is that the types of searches we have are divided up into categories. And we have the bulk, or a big portion of our searches are focused on returning genes. And this entire section here, search for genes, right? This returns any result, any search you run from this list will return lists of genes, right? And so if you expand all, for example, it shows you all of these different searches, and each one of these will return a list of genes when you run the search. So just like we, if I wanted to look for secreted proteins, so under protein targeting localization, predicted signal peptide, if I click on this, I run the same search we just ran in the previous search, and what happens is that will return a list of genes. Okay. If you're not sure where a search is, we have this tool called Find a Search. So I can start typing signal, for example, and you'll see here it filters my searches to signal peptide. If I was interested in transmembrane domain, you start getting the transmembrane domain. If I'm interested in structure, for example, you just start typing struct, and you'll see that anything that has the word structure, any search that has the word structure will filter and show you right here. Okay. There are other searches that we make available. So these are not gene searches, but they're what we call search other data types. So for example, you can search all the isolates that are available in PubSet. So if you're a population, if you work on, if you do population biology, and you're interested in surveying what kinds of fungal populations we have based on, these are typically based on single or multi-locus sequencing, so not high throughput sequencing. So this is not looking at whole genome polymorphisms. It's simply looking at typing of isolates based on a single locus. And so you can run searches here, and these will not return genes. This will return the actual isolates themselves with the information about the isolates. And you can see here, you can search for isolates based on geographic location or based on taxon and so forth. So for example, I can click on taxon, and this will load all the various Plasmodium, in this case, species that are available from the database. Or I can go and say, well, I want to look for isolates that are deposited in the PubSet database, which is a GenBank database, that are from Brazil. So we can go ahead and type Brazil here, and then this will filter it out, and then you can run this and get answer. And so now, instead of returning sets of genes, this is returning sets of isolates. So there are 3,851 isolates that came from Brazil that are available in the PubSet database, and you can search them here. You'll notice here that I have two lines here, two strategies, and that's because I ran a different search strategy before, and which I will demo in a couple of minutes. But basically, every new search will come up in a new search strategy, and if you add steps, you grow this expanding search strategy. So another, as I was mentioning, let's see, for some reason this, there we go. I don't know why that's happening. So as I mentioned, so we have the population data here, but you can also search for genomic sequences. So again, if you're looking for contigs on a genome, you can look for genomic segments. So you can, for example, use a DNA motif pattern and search for particular DNA motifs. So if, let's say you have a sequence, nucleic acid sequence for transcription factor binding site, you can run the search here. You can look for polymorphisms based on isolates that are available in the database. We will demo a SNP query or some of the SNP queries later on today. There are additional searches. You can look for metabolic pathways. Again, this is another aspect of the resource we won't explore today, but you can quickly, for example, go in and search for a pathway based on its name. There are many different sources of these, but I can, let's say, go in quickly and say I want to look for glycolysis, gluconeogenesis, glycolysis right here. You can click on Get Answer, and then this will generate a glycolysis page, basically. So again, we're not returning genes, but we're returning the entire pathway. And here's the glycolytic pathway. You can zoom into this, and as you zoom in, the structure of the metabolites will show up, and then zoom in more. The enzymes with their EC numbers will show up here. The ones in orange are ones that were found in the database. So this is glycolysis, so not surprising that there should be a representation of most of the enzymes in glycolysis, in the glycolytic pathway. You can, one thing that's pretty cool, and again, I will just show it to you here just for the sake, so that you know, but you can actually paint the pathway based on various things, including experiments or, for example, genera. And so you may want to say, well, I want to find all, show me if there's any difference between the different fungal organisms that we have here. So, so I'm adding, I think we only have 3 right now, but we can, oh no, we have more. They're just not organized appropriately. So let's just go ahead and pick some of these. I think let's put Raspberry as well, and let's put some of the Microsporidia. Okay, and once you click on Paint, what it does, it'll repaint this section, or the section of the pathways that have the enzymes, and it'll actually, as you can see, the graphs are showing up in the background here. And let me click on one of these. There we go. And then once this loads, it should show you a more detailed graph, which shows you the distribution or phylogenetic distribution of the gene in the various taxa that I've selected. Let's give this a second. All right, so this will hopefully load in a second. All right, so while this is loading, as you can see, I think lots of people, oh, there it is, it loaded finally. So as you can see here, basically this particular enzyme, hexokinase, it looks like it has a representative in the various Microsporidia, and then it's in Aspergillus and Fusarium and Coccidioides. But then for, I must have picked Trichomonas here, so Trichomonas does not have that enzyme, for example. So, so this is a quick way for you to see potential missing biological functions in various species. So I can now, once you look at one of these graphs, you can quickly now see here that it looks like one of the Microsporidia is missing. So if you click on that, and it looks like, yeah, in Nematocida, for some reason, the gene, an ortholog of that gene is missing. Now, it does not mean that it's not there. It could be an issue with the annotation, but at least it gives you a starting point to start investigating this further. Okay. So I'm going to quickly go back to my previous strategy, and I'm going to turn it over to Eve, just so we can cover other aspects of the resource. But this is my previous search. Remember, I ran a transform by Orithology. So we ran our first search where we asked for genes based on whether they have the word kinase. Then we intersected the results with anything that has a single peptide, and we came up with 51 genes. And now I asked, expand these 51 genes to the orthologs across all the fungal organisms in the database. And so now you'll see you have orthologs across all the fungi. And so that's a nice way to access this. You can add even, and so there's another phylogenetic search that allows you to identify genes based on conservation across the tree of life. And that one, you can add a step here, and I'll just demo it just so you know. If I go under here, search for genes, and if we go under Orthology and Synteny, you will see here there's an Orthology phylogenetic profile search. And if you click on this, this gives you a tree down here at the bottom, which allows you to select things that are, for example, let's say we're now interested in anything that is not conserved in mammals. I don't know, let's say we're looking for kinases that are important in, that are unique and are somehow having an, or maybe these are secreted kinases that you want to target with a vaccine, and so you don't want it to be present in the host. And let's say the host is mammals in this case. So you can pick mammals, and you can say, well, I want to find all orthology groups that do not contain mammals. So if I click on this, notice as I click on this, the icon changes. So if I click once, it means it needs to contain all mammals that are in the database. If I click twice, it means the group that's returned includes no mammals in the database. And again, now I can intersect this result. So this is going to return all genes from fungi and oomycetes that came from groups that contained no mammals in those groups. And now I'll intersect these results with my previous ones. And again, this may take a couple of seconds, but basically it will run in the background, add another step here, and give me the genes out of these 5,679 genes that are secreted kinases that have from Aspergillus nigulans that have orthologs across all fungi. And now it'll ask how many of these, it'll return any of those genes that do not have orthologs in mammals. And so we'll let this run in the background here. I'll quickly look at the this table here. Let's see if anything else we want to show here. I don't think so. I think this is good. So as this is running, so another thing you can do in, I mentioned that it's important or it's useful to log in and create an account. And the reason for that is that if you log in, all of your searches will be saved. And then you can always go back to them and run them again or pick up from where you started, so you don't have to always reinvent the wheel. And you can also share the strategies. So anytime you create a strategy and you want to share it with a group of people, you can create a link. So I'm just going to move this pop-up window here, and you'll see there's a panel here that gives you access to the strategy and allows you to interact with the strategy in different ways. So once I turn it over to Eve, what I'm going to do is I'm going to log in the background, and I will save this, and I'll share it, and I'll put it in the chat window so everybody can have access to it, and then we can also share it with everybody else by email. So you'll get the same exact strategy I ran here, and you can open it in your own workspace. So Eve, before I turn it over to you, I'm going to just pause for a second and give people an opportunity to ask questions. So feel free to type your question in the question panel, and we'll look there. If you want to speak and ask a question verbally, then you can click on the raise hand icon, and we'll see you, and we'll unmute you so you can speak. As we wait, you'll see here in the background that search ran, and so it tells me that 2,600, about 2,600 of my genes across all fungi do not have orthologs in mammals. And again, you can go down and look at them more carefully. And again, you can filter down to whichever organism, but it looks like there are about 40 of them in Aspergilli that are secreted kinases that have orthologs across fungi and also do not have orthologs in mammals. Okay, we'll pause now for maybe just 20 seconds or so and see if there are any questions. Okay, I think I don't see any questions popping up. Again, just feel free to add them throughout the webinar, and we will be happy to answer them. As we're waiting, I'm logging in just as myself. And so you'll see here now I'm logged in. You'll see here that I have this tab here shows that there are 537 strategies that are in my history. So this is a nice way to go back and access them. Let's see, it looks like there's a hand raised, I think. Nope, I don't see it. Okay. Okay, so I can give this a name. So I'll call this I can save it, and we can always then, and now if I wanted to share it with all of you, I can click on share. It generates this unique URL. So I copied this, and now I can put this in the chat window. And so I will chat this to the entire group, but also we will email it to you afterwards. So it looks like there's a question, and the question is, in the orthology phylogenetic profile, is it possible to compare synteny between different organisms, such as the order of genes is different in species? So you can ask for orthologs. So in this step here, where you transform by orthology, I can click on it, and that's actually a nice way to demo that once I mouse over this panel with my mouse, you'll see here that there's a little edit icon that appears over almost everything here, which allows you to go in and revise any of these steps. So now I can revise this orthology step, and you'll notice here there's an option for you to select if you want to only return synthetic orthologs. And by synteny here, we're talking about collinearity, obviously. And so that's I think that's the way you would be able to do this and run this. Obviously, once you do this, I can run the step. It may take a few seconds, but in the background, this number of orthologs should, in theory, go down dramatically. Well, I don't know how dramatically, but it should go down at least. And so we'll let this run in the background. But that's basically how you would go about doing that. Okay, I hope that answered the question. So yeah, you'll see, so the number went down by at least 4-fold, right? And so now there are 1,500 genes, and again, a lot less genes that do not have orthologs in mammals, as expected, basically. And when we visit a gene page later on, you'll see we have also a synteny view that shows you the synteny. There's a question about particular fungal species and where they're available. Eve, do you want to address this one? This is, I don't think we have, so again, it depends on so we obviously don't have all organisms, all fungal organisms. Our funding is mostly geared towards pathogenic fungi, although we clearly load more than just pathogenic fungi. So we try and load as many evolutionary relevant organisms as possible, because those turn out to be very important even for people working on pathogenic fungi. And if they're pathogenic fungi, obviously there's a priority list of which ones we work on first. But Eve, I don't know if you want to address this question in particular about those specific organisms.
Yeah, for fungal organisms, you normally would navigate to the FungiDB database. If we do not have a genome that you're interested in, and it looks like we do not at the moment, do send us a note. You will notice in the handouts that we posted that there is a link, nominate a dataset page. This is a page that you can communicate with us by suggesting various datasets that we should integrate, especially if these are model organisms for medically or any other relevant fungi. And all of these datasets that are being submitted further communicated to the advisory groups who help us to prioritize and schedule the data.
And if you're ever interested in knowing which organisms are in FungiDB currently, and so what I did is I just navigated to identify genes based on organism, and it gives you a table here, which is sort of broken down into different organisms. But also on the home page, there's another table that includes all the organisms. So there are different ways of looking at them. I like looking here because I can quickly filter, right? I can start typing and sort of finding where my potential organisms of interest are, right? And so try it there. If you don't find them in here, then just go ahead and send us an email. Let us know about the organisms you think should be loaded in FungiDB, and if you give us a rationale why. And also, obviously, the genome needs to be available somewhere for us to load. We don't do any of the sequencing ourselves. It's all done by the sequencing centers, and the annotation is done by mostly. We do have some organisms that we annotate in-house, but mostly are done by the sequencing centers. And so that's something to keep in mind as well. OK, great. Any other questions? OK, so I think now I will Eve, I'm going to make you the presenter. I think I have to do it. There we go. And so now, Eve, whenever you're ready, you can start your portion.
Okay. I am sharing my screen now, so let me know if you're not seeing it. So let's see. Okay, so as Amar just showed you a few minutes ago, you can create search strategies to analyze the underlying data in our databases. And in a few moments, I'll walk you through the RNA Seek search using a fold change, looking for the fold change in a particular dataset, and show you how to navigate to it. So if you are elsewhere within the database, not on the main page, one way to access the transcriptomics search would be to click on the new search, genes, because you're looking for differentially expressed genes, transcriptomics data, and then click on the RNA-Seq evidence submenu. If you are navigating to the search from the main page, the transcriptomics menu is located within the search for genes box, as we have shown before, and I would be interested in creating a search using the RNA-Seq evidence. So by clicking on the transcriptomics menu, the menu expands and provides an option to select an RNA-Seq evidence submenu. When you click on the RNA-Seq evidence submenu, will take you to a new page, which is an interactive table that lists the available data sets, and this is quite a nice list, as well as the type of the data sets available, which is on the right, and also the organism. It's basically the genome against we mapped raw reads. There are also the type of the searches that are listed on the right. I also explained a little bit at the top as to what they are. Notice that there are 5 different options that you can use, and I will go briefly over 4 out of 5 and then demo how to perform an RNA Seek search using fold change option. Okay, so similarity search basically allows you to find genes that have similar expression profiles to your input gene. When you use a similarity search, it will ask you to input a gene ID that you are wishing to essentially match to other data sets and find similar expression profiles. We have 2 I believe fungal data sets available for this search at the moment, and we are planning on expanding more of adding more data sets. So the next search, DE, or differential expression, this search is based on analyzing RNA-seq data via DE-seq2 analysis pipeline that takes into account experiments with replicates. So in order to run this pipeline, you need to have at least 2 replicates. So all of the searches that have replicate data reported from the paper would have this search showing up as next to their title. The percentile search is essentially a search that allows you to rank your genes based on maximum and minimum expression percentile values. For example, using percentile search, you can find highly expressed genes within the sample that are within 80 to 100%. Followed by a sense anti-sense search, which is a new feature that we rolled out in the release 40, and this feature takes advantage of the stranded data. So it's essentially allows you to look for simultaneous changes in the sense and anti-sense transcripts relevant to the reference sample. The specific application of this type of searches, for example, you could think of looking at the data when you might expect that the anti-sense transcripts may be a part of the regulatory network. So you could potentially look for the genes that, let's say, at the growth stage X, have an increase in the anti-sense transcript, when compared to the time point 0, for example. The last search that I will actually demo during the presentation is a fold change search, and this is essentially a pairwise comparison of the RNA-Seq expression data, where I use the FPKM values reported for the dataset and look, for example, at upregulated genes 2 or more fold when compared to the reference sample. Okay, so this is just a brief overview of what these searches do. And next, let's search the RNA-Seq evidence for Coccidioides immitis. So this is quite a long list of data sets, and rather than trying to sort through all of them, I would go ahead and try to filter the dataset based on the name of the species. And here's a saprobic versus parasitic growth RNA-Seq dataset that we will analyze via fold change. So the dataset that I will show you is has a publication related to it, and this is essentially a study where they have done a comparative transcriptomics analysis of the saprobic and parasitic growth phases in C. immitis from John Taylor's lab. And notice that when you arrive to the parameter selection screen, it's subdivided into two options, or rather two sections. There's a left section right here that allows you to select the parameters of your search. And the section on the right is actually graphical representation of your search that allows you to visualize how the changes that you introduce within the selective criteria on the left will affect your search. So let's go ahead and set our parameters. So if an experiment is associated with several different samples, they will be housed here, and you will have an option to select one or the other. In this particular case, there's we're looking at one experiment, looking at the different gene expression in C. immitis. And in this case, I would choose to look for any genes that are protein coding genes. If you select another option such as all, this search will include other genes such as transport RNAs, and etc. So notice that there are several options as to what type of gene expression differences you can select. For example, you can select for genes that are downregulated, upregulated, or up and downregulated. In this particular experiment, I will choose upregulated genes. Note that the graphics on the right is now changed to reflect only the upregulated search. So as we walk through, we need to make selections for our reference and comparison samples. And in order to do this, I would want to select as a control sample, in my case, the saprobic hyphae. And I would be interested in looking for the genes that are upregulated by twofold in parasitic spherules. Know how the graphic changes to where now the expression level reference sample is listed in the blue, which is the saprobic hyphae, and we are looking for the twofold or more upregulated genes in parasitic spherules, which is the expression level of the comparison samples. Okay. So before we go ahead and click on Get Answer and to find genes that are upregulated in the parasitic spherules, I want to bring your attention to the flow parameter that is described right here. Flow parameter essentially deals with the situations where fold changes data may be misled by the FPKM values that are approaching very small values or sometimes 0. So to avoid misleading fold change calculations, we have introduced a default floor parameter that is being essentially set to 10 map reads for an average gene. This parameter changes based on the type of the sequencing data and the average transcript size. And normally, I would normally leave it as at a default, but you could potentially increase the stringency later on to see how that would affect your results. So let's go ahead and identify the genes that are upregulated. So notice that the search returns about 1930 over 1,500 genes that are upregulated 2 or more fold, and some of this parameters already familiar to you. For example, this is an output across all species, but because we search for the C. immitis, you will have all results mapped to only one organism. This is why the rest of the selection will be 0 genes. And if you scroll down, you will find the results table, which essentially lists gene IDs, the transcript IDs, gene name when available, description of the product, as well as FPKM values, and the graphical representation of the FPKM values. Remember, if you would like to save your strategy, you can simply navigate to the top, save it to your account, and then share it with others as you see fit. The accounts in the EuPathDB sites are free and quite handy when you're working with your data in the EuPathDB Galaxy, which Amar will demonstrate how to do that shortly. So, all right, we are now looking at over 1,900 genes, and there are several things that you can do with this. For example, you can download the information, and by clicking on the download link, you will essentially be presented with a few choices that you can choose to export all data associated with the dataset, as well as a plethora of other useful information. In addition, I would like to show you how to further analyze your data using a Go enrichment analysis. So I have pre-run some of the strategies. So here we are back at the previous screen, and I would like to perform a Go enrichment on my genes to see what type of genes might be enriched in my sample. So to analyze your data, navigate to the Analyze Results tab, which is highlighted in blue, and you will have an option of 3 types of enrichment analysis. You can look at the word enrichment or metabolic pathways. In this case, I will demonstrate how to do Go term enrichment. So by clicking on the Go term, you will be redirected to a next screen, and the next screen, while the other one is running, I already run this previously. So you'll essentially be offered a few choices to select, and especially which type of ontology you would like to perform analysis with. So there are several types, such as molecular function, cellular component, or biological process. In this particular case, I will choose the biological processes, both computed and curated Go terms, and we'll click submit using the default p-value cutoff. And while the search is running, oh, here we go. It's quite fast. So this is the sort of the standard output for any Go term enrichment analysis. There's a few things that you can do. As always, you can export the results by clicking download. You can also view the word cloud as to what are the most common terms that are occurring within the results returned, such as you would notice that this is a statistically significant graph where most of the genes are enriched for the phosphorylation, protein phosphorylation, etc. There's another useful thing that you can do with your terms is to look at them a little bit further and potentially also use them in the search search queries later on. So the genes that are corresponding to the Go terms listed on the left are also linked So for example, for the phosphorylation, we have found that there's 42 genes in your result enriched for the Go term 0016310. And if you would like to learn more about these genes and specifically get the list of gene IDs that correspond to this number, just click on the letters in blue, and you will be redirected to the new screen with essentially gene IDs being listed as a results table. There's another thing that you can do is actually to look at your results in Revigo by opening your by essentially redirecting your page with the results into the graphical output for Revigo. And I have pre-run the search already, but essentially by clicking on the button, you'll be redirected to the Revigo site. And when you click on submit the p-values as well as Go terms that are being passed on to the site, it will generate a nice visual output of what type of terms are enriched to your analysis, as well as provide additional statistics. Notice that the Revigo provides provides you an option to collapse the Go terms and display only parental terms, which are quite nice when you're trying to take a big picture and see what type of enrichment you are observing in your samples. Okay, so showed you Revigo, and let me just show you before I'll pass it back to Amar. I'll show you how to export the genes in list and the Go term enrichment analysis into a strategy. So by clicking, let's say, on the 42 genes that are identified to have a Go term for phosphorylation, we can create a new search strategy. So these are 42 genes that are now being returned, which are listed right here at the bottom. You can also download this list and save it, or you can save the strategy in your account, and you can also add step and create all kinds of other searches, such as look if these genes are expressed in various RNA-Seq evidence datasets that we provide, or microarray, or SNPs, or proteomics. I think this is it. Amar, is there anything else that I'm missing?
No, I don't think so. There was, I've answered a few questions while you were talking. There's one about being able to run the Go enrichment on your own gene set, and I'm happy to demo it right before I discuss the SNP queries and moving forward.
All right, sounds good. Then it's back to you, and let's maybe give it 10 to 15 seconds to answer any additional questions there may be.
Sounds good.
Let's see. I think there's a question. I'm trying to navigate to it.
So I would suggest opening the other document that embeds.
Yeah. So.
First one in there, which I copied the stuff out of the bottom. Which is
okay. So maybe we can answer them verbally. So the first question was about being able to run the Go enrichment on your own gene list, and the answer is yes, you can do that. So if you have a set of gene IDs, and so I don't know if you can see this. So I think you should be able to see my PlasmaDB homepage. We do have a search for genes based on identifiers. See right here. And so if you click on this, you can click on gene IDs. And once you do that, you get the search with a window here that you can paste a bunch of gene IDs, or you can upload a text file, or you can get it from other places. But let's say it's your own list of genes. Let's say you ran a proteomics experiment and you have your whatever 60 most abundant genes in your list, and now you're interested in doing a Go enrichment on them because you want to see if you have something enriched. So I paste my IDs in here, and you click on Get Answer. This creates a step in a strategy with 62 genes. I had 62 IDs in my list, and now I can collapse this. So I can take this and I can just scroll down and click on Analyze Results and just follow the same steps that that Eve just followed and showed us in the previous demo. So hopefully that makes sense. Let's see, Eve, did you look at any of the other questions? I know there were a couple of other questions potentially.
I think there's no questions in addition to that. At least I cannot see.
I see one more that's asking, is there any way to perform a search of DNA motif enrichment in promoter regions? So the quick answer is no. So we cannot do the enrichment, DNA motif enrichment in the database. What you can do, so what I would recommend is using a different resource to actually look for motif patterns. So let's say you have a set of genes. So let's say here are my 62 genes, and you're interested in knowing if these are potentially co-regulated by the same transcription factor. And you would like to now identify if there's a region in the upstream area of those genes that is in common between all of them, right? Or between many of them. The first step is you have to download the sequence that's upstream of these genes, and we have a tool that allows you to do this. So I'm looking at my 62 genes. As you scroll down, I'm going to go back to gene results, and you'll see here there's an option to download. And once I click on download, there's an option here to get the FASTA sequence of my results, but also notice here it says configurable. So this is very important and actually a very handy tool. And so now what I can do is I can go down and say, well, I want to download the genomic sequence, right? Because I'm looking for things that are outside of the gene. And I want to, this is all relative to whatever you select. So if I select, let's say the start codon, minus would be going to the left of the start codon, so outside of the gene, and then plus would be going into the gene. So I can select minus, and let's say I want to get minus 500. And I don't care about the gene itself, so I want to stop at the start codon, right? And 0 would be the A of the ATG, right? And so if I really want to not include the A of the ATG, I can make this into -1, right? Or if I want to, if we make this into a +2, every sequence we return should end in an ATG, right? And so let's see how this works. So now when I click on Get Sequence, I'm showing in my browser. You'll see here's the 500 nucleotides upstream of every single gene, and you'll notice each one of them is ending in ATG, right? Because I added +2. Okay, if I make it -1, they will all end in the nucleotide right before the A of the ATG. Now you can take these sequences, and I would recommend going to a resource like MEME. So if you're not familiar with this, it's a very nice suite of different tools, and one of them, MEME, allows you to identify patterns between sequences. They continuously add additional tools, so I would recommend and highly recommend using this. This will then identify the motif, and then you can always go back to our resources, and once you identify a motif, you can go back to our resources and run, for example, a DNA motif pattern under genomic segments, DNA motif, and then you can type your DNA motif right here and search for whether this motif occurs in the genome, and then you can find genes that are close to that motif. So that's a way where you can merge between the pattern finding tool and looking for a particular motif within the database. I see there are a number of more questions coming up. I don't know, Eve, have you looked at any of them so far or not yet?
Yes, just a quick reply about adding new data sets in the genomes. Yes, absolutely. We are adding genomes all the time. If you could click on the Contact Us link and send us additional information about the genomes, their location, and why you think they should be integrated, we can share this information with the advisory groups who help us to prioritize and schedule genomes for integration.
So there's one more question about whether you can feed or put quantitative proteomics analysis in order to compare with other databases in the platform. And so we do load quantitative proteomics data in our resources as a data set. And so, for example, if I'm looking in PlasmaDB, there's a section here called proteomics, and you notice there are 3 types of searches available. There's mass spec evidence. This simply looks for peptides within genes. So we align peptides that come from an analysis. post-translational modification, so you can identify genes based on PTMs, whether it's phosphorylation or acetylation, for example. And then we have quantitative mass spec evidence. And so again, these searches are very similar. So if I click on this, you'll see there, right now we only have 2 quantitative mass spec experiments in PlasmaDB, and these are fold change queries. So I can click on fold change. And it's similar to the RNA sequence query that Eve just showed you. So you select which stages you want to compare to each other. And so you, for example, identify genes that are upregulated in schizons compared to ring stages by at least 1.5-fold. Get answer, and this will return the results. This is based on a quantitative proteomics experiment.
And there's one more thing that I wanted to add, Clayton. If you have a custom set of genes that you would like to compare against the integrated data in PlasmaDB or any of the databases, you can create a gene IDs search and then add a step to bring in either RNA-Seq or proteomics data, just as we have showed previously when constructing queries.
Right. So here, for example, I loaded these 62 genes because I was interested in doing Go enrichment. I can also say, well, I wonder how many of these are upregulated in the quantitative proteomics experiment, right, that I just ran. So let's pick, I think I picked this one. And so now I'm going to say, I want to find if any of these genes are upregulated in schizons compared to rings, intersect, and run the step. And so now it tells me that, yeah, 22 of these genes, of these 62, are upregulated in the schizon stage. Okay. Okay, I think so. Please keep adding the questions, and we'll try and address all of them if we can during this webinar. But I will carry on. Eve, does it sound good? Should I carry on right now?
Yes, I will keep an eye on the questions and try to answer them.
Okay, great, thank you. So what I wanted to demo next is running a polymorphism or variant query in our databases. I'm going to be using PlasmaDB as the example. So I'm going to go ahead and close some of these searches. Just as a side note, when I click on this little X here, it's not deleting the strategy, it's just simply closing it. If I ever want to go back to them, I can go back to my all section. I'm logged in. If you're logged in, this is the advantage, right? I can click on the all section here, and it'll load all of the searches. So for example, this is one that's already open, but this is one that I ran before that I just closed, right? If I want to open it again, I can just click on it, it'll open it, and now I can go back to it, okay? If you save it, saved strategies are listed in a special section down here. So these are ones that you can actually then create custom links for and share them with other people and so forth. You can also make them public. And so we have a public strategy section. And so at any point, I can add a description to one of my tools, one of my searches, and I can make it public, and they'll show up in the public strategy section, which are accessible by anybody. So anybody can click on them and see what the search results look like. Okay, so what I wanted to do is I wanted to expand on the question that Eve was doing before. She was doing it in FungiDB. I'm just going to switch to PlasmaDB. And if I go back to my strategies, I already ran a search, which was an RNA sequence expression search. And so I looked for, I'm looking, again, there's 62 genes here in this list, but I looked for genes that were upregulated early during infection and also late during infection. So the question I'm asking here is, find me genes that are potentially important in invasion, right? Because they're expressed late and during the early stages of infecting a new red blood cell. I ran the search just like Eve did with the RNA sequence query, selecting the parameters. We're happy to run through this again if you have more questions, but with the interest of time, let's assume that we've run the search. And now the next question I want to ask is, well, are there any of these 62 genes, do any of them have a high number of non-synonymous mutations? So mutations that are causing a change in the amino acid sequence. Presumably this would indicate that they're under pressure to change, right? So they're under selective pressure to change. And so I can add a step. And in this case, I'm looking for genes, and I'm going to identify genes based on genetic variation. And depending on the database, it may have different, you may have different options here, but one option is SNP characteristics. And so I will select this one. And again, you have to, depending on which organism you're working on. In this case, I was looking at Plasmodium falciparum 3D7, and so I'm going to keep it there. And these are all the studies that are loaded in the database for Plasmodium falciparum. There are about 200 samples that are loaded. I can go in and select specific studies. So if there is a study that I'm aware of that I want to identify SNPs in, I can do it here. I can also do it based on the available metadata or information about those samples. So for example, I can specifically say, well, I only want to look for SNPs that are present. We only have 4 isolates from Brazil right now. This will increase over the next year. But basically, I can select any of these and filter my isolates based on geographic location, and then ask for SNPs there. For the purpose of this search, I'm actually going to go ahead and select all of them. I'm interested in looking for genes that have a high number of non-synonymous SNPs, regardless of where they come from the world, right? That this is genes that are under pressure to change in any kind of environment. So now I've selected all of my 202 samples in the database. Now, as you scroll down, you'll notice here that there are a number of options that come up, and it could appear overwhelming in the beginning, but it's actually quite straightforward. And each of these question marks actually provides you with additional information about how to use these parameters. But I will quickly explain this. Read frequency threshold allows you to select SNPs based on the number of the frequency of reads that aligned. And so, for example, and this allows you to look for SNPs based on whether the organism is haploid or diploid or more than that. And so you can actually play around with this and change the number. In the case of Plasmodium, it's a haploid organism, so it should be pretty straightforward. So you can leave it at the default, a minimum threshold of 80%. Again, if you imagine in a perfect world, right, if you have a polymorphism at a particular spot, a SNP, right, at a particular spot, then all the reads from that organism that has that SNP, all of them for that spot should contain that SNP. It should be 100%, right? But it's not a perfect world. There are always mistakes and so forth. So that's why we choose 80% as the lower threshold for a haploid organism. If it was diploid and you have a diploid SNP, right, a heterozygous SNP, then you would expect it to be 50% in a perfect world, right? And so, but since it's, again, not perfect, we end up then doing this based on doing it based on 40%, right? And so depending on your organism, obviously you want to play around with this parameter. I'm going to leave it at 80% because we're looking in Plasmodium, which is haploid. Percent isolates with base call. This actually gives you information about the stringency of your search. So we have 202 isolates selected, and not every position will have enough reads, good enough reads to be able to make a base call at that position, right? And so you can choose to increase this number, and you could say, well, I want to make sure that at least 50% of my isolates have base calls at any position where I'm calling a SNP that I'm returning in my search, right? So you're increasing the stringency. So you're looking for something that's more, that you have more confidence in that it's present in all the SNPs. You can obviously increase this to make it more stringent. And then the class of SNP, if you click on this, you'll see here there are different options. And I think all of these should be intuitive. Nonsense would be stop codons. And then in our case, we're looking for non-synonymous SNPs. So I'm going to select non-synonymous. And now I can select how many of those do I want to find per gene. And let's go ahead and select 20. So we want genes. We want to return genes that are haploid, that have haploid SNPs, that have a base call in at least 50% of the isolates that I've selected, and that have at least 20 non-synonymous mutations. Okay, and then there are other parameters here that I would encourage you to explore different ways to sort of cut the pie, basically, depending on what you're interested in. And in my case, again, remember my previous search was looking for genes that are upregulated in schizons and early rings. And I want to ask how many of them have at least 20 non-synonymous SNPs when I compare all the isolates, right? So it found 2,000 genes that met the criteria in all of PlasmaDB, so that's a good number, but only 24 of them, so about a third of the genes that are expressed at those stages are also highly polymorphic and have non-synonymous SNPs. So I can scroll down and take a look at them, right? And I think one word of advice I would give anybody using any database is that you should always, you're the scientist, right? And so just because it's showing up in a database, it doesn't mean it's accurate or correct. So you always have to go back and look at the results that you're getting and ask yourself, well, do they make sense? Are these results that you would expect to come back in your gene list, right? And so luckily, I do have things in here that make sense, including this erythrocyte binding antigen. There's a MSP6. These are genes that are known to be highly polymorphic, and also some of them are known vaccine candidates, or at least highly antigenic. And so that's not surprising. And so that's the description of the how you can intersect with SNP queries. We have a number of other types of SNP searches. So again, I would encourage you, if this is something you work on and you're interested in finding more about this, send us an email and we can send you more details, or potentially chat with you more about all the different types of SNP queries that we have available to you. I'm going to pause for a second before we take a look at gene pages, because I noticed there are a couple of questions showing up here. So okay, so there's a question about being able to draw a phylogenetic tree on a specific gene. And Eve, I think I will switch back to you to talk about gene page, and you can maybe specifically demo the drawing the similarity tree, at least just showing the multiple sequence alignment on a gene page, because that will address one of the questions that came up.
Yeah, I think the questions from Hans and Kunal, both about phylogenetic trees. So I can demo it when we cover gene pages tools.
Okay, so would you like me to switch to you to do gene pages right now, or do you want me to continue with RNA sequence? How about we do the gene pages, and then we'll switch to RNA sequence. Does that sound good?
Yeah, sounds good. Let's do that.
And of course, people continue asking questions.
All right. So let me switch screens.
Do I need to make you presenter, or are you able to switch?
I think I should be able to. Here we go.
Okay, perfect.
All right, we are back to FungiDB. Go ahead.
I was just going to say, and I think it's great that people are asking so many questions, so please continue adding them in there. That's very useful for us to be able to see all these questions, and we're very happy to answer them.
Yes, absolutely. All right, there are a few features, new features that we have added quite recently, and I will show you how to navigate to 4 of them. And all of these tools are located within the gene record pages, which means when you click on the gene ID, let's say I will type in what would be a good ID to do. Let's, for example, take a look at the Fusarium graminearum gene. Here we go. So all of the tools that I will talk about also have individual tutorials associated with them, which can be located if you navigate to the release 40 news. And the searches that I will talk about is the Clustal W alignment, BLAST-P, MITRE-PROT, and WOLF-P sort. All right, so going back to the Fusarium graminearum gene, perhaps if you're working on mitochondrial proteins, you may be interested in looking for genes that have sequences that target them to mitochondrial organelles. So one way to potentially figure out if this could be true, or at least get an idea of how well it matches against the available data, you can navigate to the analysis of sequences, which is a sequence analysis menu within the contents menu on the left. And I will go ahead and click on the link and scroll down to, actually, it's protein features and properties. Apologies. So in the section Protein Features and Properties, we have added several options. You can blast the protein sequence against 8 known databases. For example, if you take the sequence for this particular gene that we're looking at participating in the phosphorus metabolism, you can select a non-redundant protein sequences to identify genes that are orthologous in other species. Another useful tool is actually to look at the proteins, for example, that I just mentioned that localize to mitochondria. So MitoProt tool, it's a MitoProt2 software that predicts mitochondrial-related proteins by calculating the N-terminal protein region that can support the mitochondrial targeting sequence, or MTS, and the cleavage site. So when you click on Submit, we actually transfer the sequence information to the MitoProt database, and it gives you an output of whether it is a good match or the probability for the export to mitochondria. And there's a help information available on their site to help you understand what our individual scores are, but essentially the probability that it's 0.003, the likelihood of the protein being exported to mitochondria, it's quite low. So let's see. Another tool that I wanted to show you is WolfP Sort. Just really quickly going back to the same gene record page and scrolling down a little bit more within the protein features and properties section. There's a WolfP Sort analysis that is essentially helps to predict the subcellular localization sites of proteins based on their amino acid sequences. And you can use any of the 3 databases which the WolfP Sort is trained on to look for various proteins in other species. The question that I would like to address in a little bit greater detail next deals with creating phylogenetic trees. So when we look at the Orthology and Synteny submenu right here, I'll go ahead and click on it, and brings us to an interactive table that essentially gives you an output of all orthologs and paralogs that we detected for this particular gene. And notice that there's quite a few listed. So what you can do, you can either select all of them if you want to use the sequence for the alignment, or you can select only a particular sort of subgroup of species. For example, we are currently in the Fusarium graminearum genome, and let's say I would be interested in taking all other Fusarium species, and I will go ahead and create a search to select all Fusarium species except Fusarium graminearum. So I'll go ahead and scroll down and select check all and deselect Fusarium graminearum in this case. So basically what I want to do, I want to align this particular sequence to all other sequences from Fusarium oxysporum, Verticilioides, Vergicoroi, and etc. So I'll go ahead and run the search. The alignment will be done by the ClustalW algorithm, which generates an output in sort of in 2 different forms. The first output, and that's the standard output, you essentially see the alignment of the protein sequences and how well they match up to each other, which you can export as you like. But if you scroll down to the bottom of the page, there is also a section that is called a DND file. This output can be used to create phylogenetic trees in other softwares, or in this case, I will demo iToll website to create an interactive tree that you can modify as you see fit. So let's go ahead and copy the DND output and then navigate to the iToll website. And it's fairly simple. All you need to do is copy your text, paste it in the window, and click upload. And the website will generate an output. Here we go, that you can view and also adjust using the control panels on the right. Are there any questions so far?
I think one important thing to note, obviously, from this is that it's not. You have to be careful about, calling this a true phylogenetic tree, right? So I would, I would again use this as a guide tree or a similarity tree, as a starting point. So if you were going to truly try and infer phylogeny, you would probably want to include additional things in this tree. But as a quick way to look at similarity, I think this is quite nice. The second thing to add is that, as Eve demonstrated, we're currently using ClustalW as the tool for alignment. This will actually change in the upcoming release, and we'll be switching to using Clustal Omega, which is supposed to be slightly more, more robust in general, and in particular when you're doing a large number of alignments. But basically that'll be a minor change, which I think is a positive change.
Yes, and note, as with any resources, we try to provide comprehensive data, but we obviously do not include all the genomes for all genera that are there across fungi or Plasmodium species, etc. So obviously there are some limitations. So yeah, keep in mind that you may want to look up and to see if there's any other data that coming from the newly sequenced genome, etc. when you create sort of a comprehensive tree.
Great. Okay, so the last thing I think we want to demo. I know I think we said we're going to go for an hour and a half, but I think we can go a little bit longer. I'm fine going a bit longer if people are still happy to stay on. If people have to leave, obviously we won't be mad, but we will continue talking, and we will save the webinar and then make it available. There's really one more thing we wanted to demo and make sure we touch on for this webinar, and I'll go ahead and take back controls. There we go, show my screen. Okay, so before showing that, I just quickly on the gene page. So Eve was on a gene page in FungiDB. I'm looking at a gene page in PlasmaDB. So again, the gene page will include a huge amount of information about the gene itself. Basically, everything we have in the database that pertains to a gene will be displayed in some form or another on the gene page. And the content here, the category section, allows you to quickly navigate to the different sections. But in particular, there was a question about synteny earlier, and we do display synteny on the gene pages. Actually, there are a couple of things that we should highlight here that we display on the gene pages, which will be relevant to your particular organism. So right at the top, we have a section called Gene Models. And this section will show you the gene itself. So this is the gene. It's going from left to right. And it's a single exon gene, so it has no introns. So depending on the organism of interest, of course, if there are introns in there, you may want to see what kind of evidence there is that supports those introns, so real biological evidence. I've had people come up to me, and say, well, you guys have this annotation on gene model in FungiDB or in PlasmaDB, and I spent all this time, trying to clone the gene, and it turns out it's actually a 2-exon gene, not a single exon gene. The annotation sucks. What's going on? And they're right. The annotation is not perfect. There are a lot of mistakes in the curation, right? This is inherent not only in genomes that are in EuPathDB sites, but in all genomes. This is an ongoing problem, and it's improving as we have better and better technology that assess and allow you to look at gene structure, in particular RNA sequence data. And so what we are doing is, because we don't have capacity to re-annotate and recurate all of our genomes, what we do is we map any RNA sequence data we have in the database, we map back to the genome, and then we generate information about splice junctions, which is shown here in the splice junction section. And so I remember this one example, a person really complaining about a particular gene, and on the gene page itself, if they just took the time to understand this section here, they would have realized that, oh, their gene model is probably incorrect in the annotation, because what the data is showing is that the gene model should have had two exons. This particular example is not that exciting because it's a single exon gene, but you could see here that there are little lines here which indicate potential splice junctions. And if you mouse over this, you will get information about how many reads span this particular proposed intron based on the RNA sequence. So there are 170 reads. It's not a huge number, but it's not negligible. It's not zero, right? And so, so it is, you may argue that maybe there are forms of this gene where there is an intron here and potentially another exon present up here. But again, the number is very low. Typically, depending on the number of experiments we have, you'll find the numbers, the splice junctions to be in the thousands, right? And so that's something to keep in mind. Okay, so that's all I was going to say about the gene models, but we do have information about gene models. And again, if you're ever concerned about your gene model, don't just rely on the actual official annotation gene model, but also look into the available data. And that's what we try and provide here on the gene pages, all this data that you can assess for yourself. The second thing that I wanted to quickly highlight is that we do have a section, as Eve indicated, she was looking at the orthology section right here. But if I close this and I scroll down a little bit more, you'll see here that there's a synteny section. And so this shows you the collinearity of the genes in this particular region, comparing all the Plasmodium species in this case. And so you can see that they're all fairly syntenic. And so that's not too surprising. Any of these things where it says view in genome browser, this allows you to open up another window, which is the actual genome browser, where you can browse the genomes and you can zoom in and zoom out and look at larger or smaller areas to look at collinearity. And so that's potentially of interest to take a look at. And then one more thing to highlight on a gene page is that we do have also a lot of information about functional data. So for example, this section includes all the transcriptomic data available in the database. Okay, and so these are all experiments that we have preloaded. The proteomics section also includes proteomics data. So if there was, so for example, there are a few studies where there were peptide hits, just a very few. So there's one peptide from this study, another peptide from this study, but this one was a lysine, which was acetylated. Actually, it has an acetylation site, and so there's a post-transition modification here, there's additional information on the page. Okay, so the next thing I wanted to switch to is quickly demo the tool for analyzing your own experiments, and this is using the Galaxy platform. Again, Marcus is very familiar with this, and so I'm sure he'll be happy to answer questions offline, but also I'm sure other people in the group here have used this before. But in order to use this tool, you have to log in, because this is actually storing information in your own user workspace, so the login is required. But once you log in, you can click on Analyze My Experiment. This will take you to our Galaxy site. So Galaxy is a suite of online web tools that allow you to run various bioinformatics tools using a web interface, right? And we didn't develop Galaxy. There's a Galaxy consortium that has been actively developing this tool for many years now. Anybody can actually install a Galaxy instance on their own machines. It's heavy, it requires somebody to manage it and manage the installations. But what we did is we actually have our own installation of Galaxy. It's managed by a group called Globus Genomics. We don't even manage it ourselves, because it does require a certain expertise, but also the tools, the bioinformatic tools, are going to be running on your data, and we don't run them on our local servers. They get run on the Amazon cloud, right? So we farm this out, and we pay a fee, obviously, for this, but then it gets farmed out to the Amazon cloud, and the results are run there. The nice thing about our Galaxy instance is that we've preloaded all of our genomes. So all the genomes in FungiDB have been preloaded. So you don't have to load your genome, assuming it's one of the ones that are in FungiDB. You can just select it from the drop-down menu when you run these searches. So as we indicated in the beginning, I'm not going to run through a Galaxy exercise itself. There are many tools out there. We have a number of exercises available to you, which Eve has linked in her handout, or we're happy to share with you afterwards. But just to show you a new feature that we've added that allows you to now, after you've run an analysis in Galaxy, to take results from Galaxy back into our EuPathDB databases to ask additional questions about the data. We have the first iteration of this. You have to use a particular workflow. So this is a workflow right here, which allows you to export RNA sequence data. It relies on running cufflinks to generate expression values, so the FPKM values of for your genes. This workflow also generates a file called the bigwig file, which includes essentially a coverage plot for where your reads aligned. So on the right-hand side here, I'm showing a completed workflow. So I got some RNA sequence data from the sequence read archive. I ran it through this workflow right here. It generates a bunch of output files. Each one of these is an output file. And you'll notice here there are some bigwig files, and there are also cufflinks output files. Now, once the workflow is completed, when it's all green, I can go here to the left and click on EuPathDB export tools and select RNA sequence to EuPathDB. Once I select this, it provides you with a window. You have to give this a name, my experiment 1, whatever you want to call it. You select the bigwig file. In this case, it's a collection of bigwig files. And then you can select the gene expression file, which is cufflinks output. You have to fill these up with some information here, and then you click on execute. Okay. Once you click on execute, I won't do it right now. It takes a couple of minutes to do that. So to save some time, I already ran this beforehand. You will get a little another step in your history that's called export to EuPathDB. This will run. And once it's completed, then you can go back to PlasmaDB. So I'm back to this gene page. And now, instead of going to analyze my experiments, I'm going to go to my data sets. And you'll see here it's called beta, because it's still in beta. We're still adding new features and functionalities to this. And this will, hopefully, over the coming year, will become more and more integrated. But once I click on my data sets, it will show me, once it loads, it'll show me all the data sets that I have transferred from Galaxy in the past. And so you'll see here there are 3 data sets that I've transferred. One of them called differentiation, which is the one that we were just looking at. Okay. Actually, I transferred this about a month ago. So I'm going to go ahead and click on differentiation. And this takes me to a data set page. Okay. So again, this is my data set, my analysis. And so I'm looking in here. It shows me that there's information about the experiment. I've shared this with one of the, Christina is one of the people who's been working on developing this. So I've shared this with her. So you can actually click on this share icon and share it with specific users of the database. And you'll notice here that there's an option here for available searches. And I will show this in just a second. But also, as you scroll down, you'll notice here there's a section that tells you about its compatibility. And so this is a Plasmodium burgii dataset, and it tells me that it requires minimally release 32 of the database. So even though we are currently at release 40, as you see up here, but the genome itself is still a release 32 genome. The genome itself has not really changed. And so as long as the genome is the same, this should remain valid. If we change the genome, you will get a note here saying that you need to rerun your analysis because it's no longer valid according to the version of the genome that we have in the database. And you will notice here that there are a number of lines here that allow you to transfer your data to gbrowse. The ones that are end in BW are ones that allow you can actually visualize in the database. And so if I click on view in gbrowse, it's going to load or open up another page. I will let this go in the background. Sometimes this takes a while to load, but that's going to load the coverage plot of my bigwig file or of my results that I was looking at that I generated from my RNA-Seq pipeline. Actually, this loaded quite quickly, and so I have some other things here loaded. So I'll close these. But this is the genome browser. And this is the coverage plot of my actual data. So these are the actual coverage plots. And so I can, if I wanted to, I can zoom into this area and take a look at it in more detail. But really, what's more useful is if I can go back and start asking questions about my data, right? So I want to find out from this particular experiment. I think I compared male and female gametocytes of Plasmodium burgii. I would like to find out if there are genes that are differentially regulated between these two samples. And so what we can do is we can use this available searches link. So I'm going to click on genes by RNA sequence. And so what this does, it generates a page which looks just like the RNA sequence page that Eve showed you or demoed to you earlier, but this is on your own data. So I'm selecting differentiation 1, which is the experiment that's available to me. I want to find genes that are upregulated. Let's change this to at least, I don't know, tenfold. And let's say I want to find genes that are upregulated by tenfold when I compare the female gametocytes to all the male gametocytes. So this was an experiment that had 3 replicates, 3 biological replicates. So I'm selecting the 3 female replicates. I want to compare them to the 3 male replicates. And I'm going to change this to maximum and minimum. So basically, what I'm asking is to return any gene where the minimum expression value from the 3 female replicates is at least tenfold higher than the maximum expression value for the 3 male replicates. Okay, so this graph here on the right shows an example gene that should be returned based on the parameters you selected. So it's a nice guide. It kind of tells you that it confirms to you that you've selected the right parameters for what you are interested in. Okay, so we're comparing the things in red here, the comparison samples, to the reference samples, and that's where the fold change will be. And then you click on Get Answer. And so this tells me that there were 242 genes that are upregulated by at least tenfold in my female gametocytes compared to my male gametocytes. And now if you scroll down, you'll notice this is a list of genes, but not only that, it generates a graph representing your results. Okay, so and sure enough, there's high expression in the females and low expression in the males, so that's good. So you can go down and look at them individually. You can do a Go enrichment on these, right? So these are results from my experiment. So I can quickly click on Go enrichment, and we can select whichever one of these. Again, you've demoed this, so I don't necessarily have to go through the whole motions of the entire search. But this will ultimately generate a Go enrichment for you here. Then if you go back to the gene results, actually, let's take a quick look at them and see if there's anything interesting. So it tells me here that there's a high enrichment in the female gametocytes of genes that are involved in the pellicle and the subpellicular microtubules. So structural genes, and the female and male gametocytes do look different. They do have different structural components, and so maybe this is not too surprising and potentially interesting. So going back, I will look at my gene results. So I can go back now, and as you would normally, I could visit any of these gene pages. So I'm just going to click on one of these genes. So it loads the gene page. Now the exciting part, again, another exciting part about this tool is that there's in the transcriptomics section now, there are all the data sets that we've loaded for Plasmodium berghei, but you'll notice down here, there's the actual your own data set. So we integrate your own experiment in your own private workspace. So now you can quickly say, oh, I wonder how this compares to other experiments in here. And it turns out this experiment of female and male gametocytes, we've also loaded as a data set in the database. So we can see and confirm that based on our analysis, which actually used differential expression testing using DE-Seq2, we can quickly look here and say, oh yeah, look, this gene is upregulated in female gametocytes compared to males, and they also did the asexual stages in red blood cells, and it looks similar, so I'm getting similar results, so that's reassuring. But I could obviously easily ask whether other experiments that are loaded from other groups have a similar results, and you could see that this gene is highly upregulated in gametocytes in general, in this particular experiment compared to the other stages. And so all of these are just a nice way for you to do some cross comparisons between different types of data, but comparing it to your own primary analysis. Okay, so we're approaching, I think, the 2-hour mark. And I think what we will do is maybe, unless, Eve, there was anything else you wanted to definitely cover before we open up to final questions and then end this webinar, I will I suggest that we give people a few minutes to ask questions, and then we can end the webinar. And as I mentioned earlier, we will make this available as a video afterwards, so people can watch it or share it with others.
Sounds like a plan.
Okay. So we'll take a look at the question section. So there's a question about whether there's a way to search for gene orthology on genes that are not available in the database. So there is, and so what you can do, you obviously will not do it in this database here. We have So the way we generate our orthology is based on an OrthoMCL pipeline, which is also a tool that was developed by the larger EuPathDB group, but also we maintain a database called OrthoMCL, which includes information about orthology. So I'll just go to orthomcl.org, and you'll see here, this is a database that includes phylogeny or orthology groups from across 150 different organisms across the tree of life. So we're sampled bacteria, archaea, and eukaryotes. And what you can do is, if you were looking for orthologs that are, or orthology groups that had similarity to, let's say, a single protein, then what I would do is I would suggest using a BLAST tool. And so you can blast against the groups in the database, okay? The other option is, if you have an entire genome, you can actually assign every single protein in your genome to orthology groups using the assign your proteins to groups. And what we did is we implemented actually a Galaxy workflow that allows you to do this. And so again, you'll click on this to go to the Galaxy server, and then here you will notice there's an OrthoMCL pipeline. And if you click on this, you would have to upload your proteome. Obviously, it's a FASTA file of all your proteins, and then you just simply I think all you have to do is there's nothing you need to change in these parameters, but this will you just simply select the file, the FASTA file, and run your workflow, and this will then generate an output that includes mapping of all your proteins to the orthology groups. So I hope this answered your question. Again, if it's for a single gene, I would recommend going to OrthoMCL and simply blasting the sequence against the database. So right here, and this will return all the groups that were similar to your protein. And if you are looking at an entire proteome, I would suggest using our Galaxy workflow, which maps to all the orthology groups in the database.
And just a quick note, if an organism is not available in the OrthoMCL, you can always use NCBI BLAST or PSI BLAST that helps you to identify distant relationships between proteins.
Okay, so I think, let's see if there are any other questions. So I don't think there are any other questions. So Marcus, you're muted right now, but I think you can go ahead and unmute yourself if you want. But just maybe check with you and see if there are any questions from your end or any comments.
Well, I think pretty much everybody has their laptops connected, so they're like doing questions, and I will stay here for a longer time. So if I can, like, maybe can take some questions from the folks from here. So, but honestly, it was great. It's really there was a lot of tools that I didn't know before, so it was even really good for me. Actually, I would like to thank you guys again, for you and Eve for your time, and we really appreciate that. And please please share the YouTube link, because I think that's going to be very enjoyable for everyone, because sometimes, I think still English it's a barrier for us here, but if you can, like, hear that part like 10 times, we can get it. So
sure, absolutely.
That will help a lot.
We'll definitely do that. And as I mentioned before, if anybody on the call or in the rooms has additional questions, I know people are sometimes hesitant to send an email just because they're worried about whether their English sounds good or if they're writing things correctly. I would say don't worry about that at all. Just write the question, send it, and if we don't understand it, we will ask you to explain it, right? And it's not a big deal. We'll figure it out, and we're more than happy to try and come up with an answer for you. I can give you a really good example where once I was communicating with a system administrator in Colombia, right? That I don't speak any Spanish, I speak 2 words maybe, and they didn't speak any English, and so they would send me a question in Spanish, and I would put their question in Google Translate. And I would get the answer, and then I would respond in English, but then put it in Google Translate to give him a Spanish output, and it worked, right? And so if you're worried, there are different ways of doing that. This day and age, language should not be a barrier. I think you can feel free to send us whatever question you want in whatever language, and we'll figure out a way to answer it.
Yeah, thank you. You guys are the best.
Great. Well, it's our pleasure, and we will, as I mentioned, we'll keep the recording, and we will hopefully see you guys in some meeting somewhere or talk to you soon.
All right. Thank you, guys.
Sounds good.
Okay.
Bye.
Bye-bye, everybody. Bye-bye.